The overall goal of this procedure is to rescue recombinant Newcastle disease virus, or NDV, from cloned DNA. This is accomplished by first infecting mammalian cells with a helper virus encoding T7 polymerase, and then transfecting those cells with plasmids encoding the minimal replication machinery of the virus under control of the T7 promoter. The second step is to co-cultivate the transfected mammalian cells with avian cells, which are from the natural hosts of the virus. The newly generated virions released from the mammalian cells will infect the avian cells for a first round of amplification. Next, the supernatant of the co-culture is inoculated into embryonated chicken eggs for further growth of the rescued virus. The final step is to collect the allantoic fluid from the inoculated eggs. Ultimately, a simple hemagglutination assay is used to show the presence of NDV in the allantoic fluid and the success of the rescue process.
Recombinant viruses rescued by this method can provide insights into different factors affecting infection and can also be used as vectors for the delivery of antigens for vaccination and antitumoral therapy.
To begin, split mammalian Hep2 or A549 cells the day before transfection in 6 well plates. The density of the cells should reach 80 to 90% confluence the following day. For each virus to be rescued, include 2 to 4 different wells. Also include 2 extra wells for the controls pCAGs-GFP and pSite-GFP, aimed to monitor transfection and MVA-T7 infection efficiencies, respectively. Wash cells twice with 1 ml of warmed PBS. Then infect the cultured mammalian cells with the MVA-T7 virus at a multiplicity of infection of 1 PFU per cell in a final volume of 200 microliters in PBS. Leave the cells to incubate for 1 hour at room temperature. During the viral infection incubation, prepare the transfection mix as described in the written text. After the lipofectamine Optimum solution has been prepared and pre-incubated for 5 minutes, add 250 microliters of the solution to the DNA tubes and incubate for 20 to 30 minutes at room temperature. Following incubation with MVA-T7, remove the virus inoculum by aspiration and replace with the DNA lipofectamine transfection mix. Add 1 milliliter of DMEM 10% FBS to each dish. Incubate the infected transfected cells for 6 to 8 hours at 37 degrees Celsius, 5% carbon dioxide. Then replace the transfection medium with 1.5 milliliters of fresh DMEM 10% FBS. At 24 hours post-transfection, control wells can be observed under a fluorescence microscope to assess transfection efficiency and T7 polymerase activity before proceeding with co-culture of the infected transfected mammalian cells with avian fibroblasts. Usually, a confluent 100 mm tissue culture dish of chicken or duck embryo fibroblasts is used per 2 transfected wells. Be sure to prepare in advance enough 100 mm tissue culture dishes of avian cells per all rescue attempts. For efficient rescue of the virus, DMEM 10% FBS medium is supplemented with 5% allantoic fluid and 30 millimolar magnesium chloride. After washing the mammalian cells twice with warmed 1x PBS, trypsinize the cells with 0.2 milliliters of EDTA trypsin until they detach. Resuspend the cells in 1 milliliter of the supplemented DMEM and transfer to a 100 mm tissue culture dish. Add 3 milliliters of the same medium. After washing the avian cells twice with warmed 1x PBS, trypsinize the cells with 1 milliliter of EDTA trypsin and incubate for 1 to 2 minutes at 37 degrees Celsius until cells detach. Resuspend the avian cells by adding 8 milliliters of the prepared medium. Then add 4 milliliters of the trypsinized avian cells to the mammalian cells in the co-culture 100 mm tissue culture dish for a total volume of 8 milliliters. Gently shake the co-cultured cells in the 100 milliliter dish to ensure a uniform distribution, and then place them in the incubator at 37 degrees Celsius for 3 to 4 days. Eggs are infected when all of the co-cultured cells are affected by the cytopathic effect due to the MV87 viral infection. At this point, remove 1 milliliter of the tissue culture supernatant and add it to an Eppendorf tube. Centrifuge the tissue culture supernatant for 1 minute at 12,000 RPM in a benchtop centrifuge to remove cellular debris. Candle the eggs using a light candling box to see the interface between the air sac and the allantoic cavity. Make a mark on the interface border, avoiding blood vessel localization. Spray 70% ethanol over the eggs to establish sterile conditions. Gently make a hole in the eggshell on the marked spot. Then, inoculate 500 microliters of the supernatant using a 1 milliliter syringe, aiming the needle vertically and directly into the allantoic cavity. Seal the nick in the eggshell with melted wax or paraffin. Incubate the eggs for 2 to 3 days at 37 degrees Celsius. Incubate the infected eggs for 2 to 4 hours or overnight at 4 degrees Celsius in order to kill the chicken embryo and coagulate the blood. Spray the eggs with 70% ethanol to maintain sterile conditions. Carefully tap the apical section of the egg over the air cavity with a spoon. Once the eggshell is cracked, remove the fragments with forceps and fully expose the allantoic membrane. Expose the allantoic cavity by excising the allantoic membrane with forceps and scissors, avoiding damage to the blood vessels and the yolk. Carefully push down the embryo with a spatula and collect the upflowing allantoic fluid with a 10 milliliter pipette. Avoid damaging the yolk membrane. Transfer the allantoic fluid to a 15 milliliter centrifuge tube on ice. Clarify the allantoic fluid by centrifuging for 5 minutes at 1500 RPM. Transfer the supernatant to a fresh tube without disturbing the pellet. Store the samples at 4 degrees Celsius for up to 1 week until checking them for the presence of NDV by the hemagglutination assay. The presence of virus in the allantoic fluid from infected eggs can be determined macroscopically by their ability to hemagglutinate turkey red blood cells. Negative control samples, such as 1xPBS or uninfected allantoic fluid, as well as positive control samples, such as allantoic fluid from any NDV virus, should always be included in any HA assay to validate it. To perform the HA assay, pipette 50 microliters of 1xPBS per well in a V-bottom 96 well plate. Then, pipette 50 microliters of the allantoic fluid samples into the wells on the first column of the plate. Perform twofold serial dilutions through the rest of the plate and discard the last extra 50 microliters from the wells in the last column. Dispense 50 microliters of 0.5% turkey red blood cells in 1xPBS per well. Gently shake the plate by tapping. Incubate the plate at 4 degrees Celsius for 30 to 45 minutes or until a clear pellet is formed in the negative control wells. Early steps in the NDV rescue procedure can be monitored, specifically the transfection efficiency and the infection with MV87. Shown here are representative fluorescent and bright field images of A549 cells transfected with PCAGS-GFP or infected with MV87 and transfected with P-site-GFP. Constitutive GFP expression driven by PCAGs is an indicator of transfection efficiency, whereas T7 promoter-driven GFP expression by P-site is a control for MV87 infection and T7 polymerase activity. The images show transfection and transfection infection efficiencies that are enough for a successful NDV rescue. After the rounds of amplification in avian mammalian co-cultures and embryonated eggs, presence of rescued virus is detected by positive wells in the HA assay. NDV induces hemagglutination by linking erythrocytes, and thus prevents their sedimentation in the bottom of the V-shaped well. Therefore, lack of HA activity can easily be distinguished by the formation of a red round pellet of red blood cells, as shown here in the negative control, and one negative result from 3 unknown samples obtained after rescue.
After watching this video, you should have a good understanding on how to rescue recombinant Newcastle disease virus from cDNA with high reproducibility.